What exactly is modified in a genetically modified organism, a GMO? It's the DNA in the genome of that organism. When you change the DNA, you're usually changing a gene. You usually want some protein or trait or remove some protein or trait. And this will result in ultimately a different function in that organism. So throughout history, humans have manipulated genes by cultivating plants with more desirable traits. That's through selective breeding. And corn is the best example because corn's early ancestor, this wild grass with no nutrition. So over thousands of years, Native Americans, through selective breeding, chose for cobs and ears, and now we have the corn that is a major food source. Genetic engineering is direct manipulation of the DNA as opposed to waiting around for crossbreeding and selecting. If there's a desirable trait, you just find the gene, cut that gene out, and put it into the genome of your organism. The easiest organisms to manipulate directly are single-celled bacteria because bacteria contain plasmids as part of their genome. These plasmids are these circular rings of DNA, and these are easily removed, changed, and replaced, allowing for relatively easy engineering of recombinant DNA from two different species. Insert a gene from whatever species you wanted into the bacteria, and this was the method used for creating human insulin. Insulin could be produced by bacteria and then isolated from bacteria instead of isolating insulin from cows. So there's an ethical issue for basically killing off the cow to get the insulin to then use for humans. And this is less of an ethical issue, killing off bacteria, which is something you do every day if you're using antibacterial soap, in order to get the protein that the bacteria has produced from the recombinant DNA. It's combined DNA from bacteria, that's the blue part of the circle, plus human genome, that's the red part. So this goes into the bacteria, and the bacteria lives its life and it expresses its own genome, as well as this little circle of DNA. This bacteria, the host cell, lives its life. It also creates this protein. It doesn't affect the life of this bacteria. Collected and grown up in large cultures in a lab, you can then collect that protein that you had introduced in. You can merge this DNA in the first place because organisms use the same bases as well as the same triplet codons. The code of different species is written using the same nucleotide bases. What's controversial about genetic engineering is the creation of transgenic organisms, where there's DNA mixed from different species due to taking a gene from one species and then introducing it into the genome of another. Like the BT corn that is resistant to this worm, but only because it has a bacterial gene inserted into its corn genome. It's also been done with rice that is resistant to viruses because it has bacterial genes inserted into its rice genome. It can also be done to make a more nutritious rice, such as golden rice, that has genes from daffodil and bacteria inserted into the rice genome to allow for vitamin A production and have a nutritious version of that rice called golden rice. The thing about genetic engineering, though, is it has its roots in good. So the original purpose is for greater good, to create better things like a rice that's virus resistant, or even in the natural selective breeding process in which corn was developed as a food originating from a wild rice. Genetic engineering can be argued that it just is more efficient and faster than selective breeding and natural processes. In creating a transgenic organism, it's not exactly like crossbreeding, though, because genes normally wouldn't transfer across species. In thousands of years, you would not have a bacterial gene inserted into a corn genome. Before deciding whether you're for or against GMOs, it's important to understand the science behind genetic engineering and knowing it's made possible by the fact that all DNA in species, plants, animals, bacteria, is all the same nucleotides. So you can easily take a part of a human genome or just the gene and insert it into a bacterial genome. Here's an example of creating a transgenic organism for a good purpose. And this is an example where you have a pathogen, which is something that causes a disease in humans, like bacteria or virus, like the coronavirus. While there are all different types of pathogens, from viruses to bacteria, humans have an advantage in which we have an immune system. Any foreign pathogens that are introduced into your body, anywhere in the cells or the bloodstream, you have an immune response that can fight off those foreign pathogens. One reason why COVID became a worldwide pandemic is because humans had no natural immunity to this brand new virus. So one method to deal with brand new viruses and diseases is to allow humans to have some exposure to them in small amounts, like in genetically modified banana like this. So in this example, there's a gene for a pathogen, which would be maybe just a portion of the pathogen, like the spike protein on coronavirus. Take the gene for the protein and insert it into the genome of bacteria. So this is that circular DNA from bacteria, and now you have recombinant DNA. That's the pathogen gene plus the bacteria gene. So two different species. This then gets inserted into plant cells, so banana plant cells, and cultured in a lab, and then taken out and grown in the fields as bananas. These grow and look like normal natural bananas, and they taste like normal natural bananas because they are, for the most part, normal natural bananas. What's different is there's that little bit of pathogen, that one pathogen protein that's expressed in the banana that doesn't have any flavor, doesn't change the taste of the banana to this person that's eating it, but that person is now a little bit of exposure to the pathogen allows for the immune response to kick in and develop antibodies against that pathogen. So it's no longer a new or foreign invader, and therefore this can be fought off if they are exposed to that pathogen or even something like that pathogen. This is something that is proposed for a vaccine instead of having a vaccine, actually allowing for humans to develop their own resistance by introducing small amounts of pathogen.